Hi friends, in this video we will discuss about the types of endocytosis. Earlier I have done a video about what is endocytosis. If you haven't watched that, go and watch it before you deal with the types of endocytosis. There are three types of endocytosis, which is phagocytosis, pinocytosis, and receptor mediated endocytosis. So let's discuss each in detail. First, we will discuss about phagocytosis. So organisms like amoeba obtain its food by the process of phagocytosis. So phagocytosis is a type of endocytosis, and through phagocytosis, intake of large particles and cells happens. So, a cell takes in large particles through a process of phagocytosis, and hence we can call it phagocytosis as cell eating. So, phagocytosis takes in large particles and even microorganisms like bacteria, virus, and even harmful substances. Now, let us discuss the steps of phagocytosis. The first step in phagocytosis is detection. So, in this step, the phagocyte, which is the cell that takes in an organism or a substance, is called a phagocyte. This phagocyte detects its antigen, that is, the substance which is provoking an immune system. So, it may be a bacteria, it can be virus. So, this antigen is being detected by the phagocyte. So, this is the first step of phagocytosis. After the detection, the next step is attachment. The phagocyte makes contact with the bacterium, that is, this phagocyte gets attached to the bacterium, and this is done by the pseudopodium, which is the extension of the cell, that is, the phagocyte extends the pseudopodium to reach the bacterium and it surrounds the bacterium, and this is how a phagocyte gets attached to a bacterium. The next step is ingestion. When the phagocyte spreads its pseudopodium around the bacterium, this bacterium gets surrounded by it, and the place where the bacterium is surrounded is enclosed within a vesicle, that is, vesicle formation occurs, and inside the vesicle there is bacterium, and this structure is known as phagosome. So, a vesicle inside which a bacterium is enclosed is called a phagosome. This phagosome is later taken into the cell, and this process is called ingestion. The next step is the fusion. Phagosome, when it reaches inside the cell, it fuses with some organelle. For example, it fuses with lysosomes mostly. When it fuses with lysosome, it is known as phagolysosome. As we know that inside the lysosome there are many digestive enzymes, and these digestive enzymes degrade the bacterium. So, in the fusion step, the phagosome gets fused with lysosome and forms phagolysosome. These lysosomes have digestive enzymes inside it, and these degrades the bacterium. The next step is elimination. The degraded material is being eliminated from the cell by the process of exocytosis. Now, let us discuss the examples of phagocytosis. As we discussed earlier, phagocytosis is performed by many protists, for example, amoeba. It is through this process that amoeba takes in its food. So, amoeba takes in its food by a process called phagocytosis. So, phagocytosis is also seen in humans. It is seen in specialized cells like immune cells. This diagram shows the process of phagocytosis in amoeba. When a food particle is being detected by amoeba, it is attached by amoeba. That is, amoeba spreads its pseudopodium over this food particle, and this food particle is surrounded by amoeba. After which this food particle is surrounded by amoeba, a phagosome is formed there, that is, a vesicle is formed there. After which this vesicle is formed, that is, the phagosome is formed. This phagosome gets attached or gets fused with the lysosomes. And the digestive enzymes that is present in these lysosomes break down the food and is digested. So, these amoebas takes in its food by a process called phagocytosis. The next type of endocytosis is pinocytosis, and this pinocytosis is known as cell drinking, whereas phagocytosis is known as cell eating. So, phagocytosis is called cell eating, whereas pinocytosis is called cell drinking. In phagocytosis, large molecules like microorganisms or large substances are taken by the cell, whereas in the process of pinocytosis, fluids and dissolved nutrients are taken up by the cell. The steps of phagocytosis and pinocytosis is almost same. That is, after which these particles, that is the substances that get attached to the cell, they are taken in by the cell and internalized vesicles are formed. And these vesicles are later transported and is fused with organelles like lysosome. And the digestive enzymes which are present inside the lysosome breaks the contents inside the vesicle. The degraded substances are removed out of the cell by a process called extracellular. In some cases, these vesicles do not fuse with the lysosome. Instead, it fuses with the cell membrane on the other side of the cell. And this is a way a cell can recycle the cell membrane proteins and the lipids. So, example of pinocytosis is the intake of nutrients in the small intestine. So, the small intestines take its nutrients by a process called pinocytosis. This diagram would help you understand the difference between phagocytosis and pinocytosis. Pinocytosis, as we discussed earlier, it is known as cell drinking, whereas phagocytosis is known as cell eating. So, in pinocytosis, it involves the intake of fluids, whereas in phagocytosis, they involves the taking of large substances. So, the steps in phagocytosis and pinocytosis is same. That is, once the particles or the substances get attached to cell membrane, it is taken in by a formation of vesicle. These vesicles move inward, that is inside a cell, and later fuses with lysosome. Lysosome has digestive enzymes within it, and these digestive enzymes degrade the substances. So, after which these substances are degraded, they are eliminated by a process called exocytosis. The last type of endocytosis is the receptor mediated endocytosis, which is also known as Receptor mediated endocytosis. So in this endocytosis, all substances are not taken inside the cell, whereas specific molecules are being taken in. These molecules first has to bind with the receptor, and these receptors are present in the cell membrane. Only after which these molecules bind with the receptors, they are taken inside the cell. Membrane receptors are seen in the plasma membrane, and these receptors are coated with a protein, which is clathrin coated pits. So once these molecules bind with the receptors, there occurs a vesicle, which is clathrin coated vesicle. And after fusing with the endosomes, these clathrin coating is being removed from the vesicles, and the contents are emptied into the cell. This diagram would help you understand the steps in receptor mediated endocytosis. The first step is that these molecules are specific, that is, specific molecules comes and binds to the receptors, and these receptors are coated with protein, and once they are bound, a clathrin coated pit occurs. The cell membrane starts to fold inwards, and there occurs a formation of vesicles, that is, a clathrin coated vesicles. Once these vesicles fuses with endosomes, this clathrin coating is being removed, and the contents is moved inside. And after this, these receptors and molecules get separated, and these receptors move to the cell membrane, whereas these molecules move towards the lysosomes. So this is the process of receptor mediated endocytosis. Now let us summarize the 3 steps of endocytosis, that is phagocytosis, pinocytosis, and receptor mediated endocytosis. Phagocytosis is the process of taking in solid particles or large particles with the use of pseudopodium, and once these particles are taken in, a phagosome is obtained. In pinocytosis, water-soluble substances are taken inside a cell, and once these substances are attached to the cell membrane, a vesicle is formed there. In the last, which is the receptor mediated endocytosis, specific molecules are taken in, that is, these molecules have to first bind to the receptor, and once they bind to these receptors, they forms a protein coated pit, and a vesicle, which is protein coated vesicle, is being obtained, and these vesicles then fuse with endosomes and releases the substances. So these are the 3 types of processes in endocytosis. Hope the 3 types of endocytosis is clear. For more such videos, do subscribe to our channel. If you like the video, please like and share with your friends.